The overall goal of these differentiation cultures is to generate cerebellar cell lineages in 2D or 3D environments.
This method can help to answer basic questions regarding neurodevelopment or disease mechanisms underlying childhood brain disorders using patient-derived stem cells. The main advantage of this technique is that it has a simple startup and maintenance with few factors, and that it can be used as a base for testing more complex neural protocols. Demonstrating the procedure will be Lisa, a technician from my laboratory.
To set up the differentiation with modified G method of passing of hPSCs, transfer the required volume of neural maintenance medium to a sterile tube, and supplement with 4 nanograms per milliliter FGF2 and 10 micromolar ROCK inhibitor. Then, keep the medium tube at room temperature or in water bath at 37 degrees Celsius. Next, aspirate the medium from the wells containing hPSCs to be differentiated, and wash the cells twice using 0.5 millimolar EDTA. In the second wash, wait 30 seconds before aspirating EDTA. After that, add 1 milliliter of PBS and incubate the cells for 4 to 9 minutes at 37 degrees Celsius. While waiting, prepare a sterile tube with 4 milliliters of PBS. Then, check the wells under the microscope. If the cells are starting to detach, free the cells by